Tay-Sachs disease is a rare autosomal recessive genetic disorder. In its most common variant, it causes a progressive deterioration of nerve cells and of mental and physical abilities that begins around 6 months of age and usually results in death by the age of 4. The disease occurs when harmful quantities of cell membrane components known as gangliosides accumulate in the brain's nerve cells, eventually leading to the premature death of the cells. A ganglioside is a form of sphingolipid, which makes Tay-Sachs disease a member of the sphingolipidoses. There is no known cure or treatment. The disease is named after the British ophthalmologist Waren Tay, who in 1881 first described a symptomatic red spot on the retina of the eye. And after the American neurologist Bernard Sachs of Mount Sinai Hospital, New York, who described in 1887 the cellular changes of Tay-Sachs disease and noted an increased disease prevalence in Ashkenazi Jewish people. Research in the late 20th century demonstrated that Tay-Sachs disease is caused by a genetic mutation in the HEXA gene on chromosome 15. A large number of HEXA mutations have been discovered, and new ones are still being reported. These mutations reach significant frequencies in specific populations. French Canadians of southeastern Quebec have a carrier frequency similar to that seen in Ashkenazi Jews, but carry a different mutation. Cajuns of southern Louisiana carry the same mutation that is seen most commonly in Ashkenazi Jews. HEXA mutations are rare and are most seen in genetically isolated populations. Tay-Sachs can occur from the inheritance of either two similar, or two unrelated, causative mutations in the HEXA gene. As an autosomal recessive disorder, two Tay-Sachs alleles are required for an individual to exhibit symptoms of the disease. Carriers of a single Tay-Sachs allele do not exhibit symptoms of the disease but appear to be protected to some extent against tuberculosis. This accounts for the persistence of the allele in certain populations and that it confers a selective advantage in other words, being a heterozygote is advantageous. Signs and symptoms. Tay-Sachs disease is classified into several forms, which are differentiated based on the onset age of neurological symptoms. Infantile Tay-Sachs disease. Infants with Tay-Sachs disease appear to develop normally for the first 6 months after birth. Then, as neurons become distended with gangliosides, a relentless deterioration of mental and physical abilities begins. The child becomes blind, deaf, unable to swallow, atrophied, and paralytic. Death usually occurs before the age of 4. Juvenile Tay-Sachs disease. Juvenile Tay-Sachs disease is rarer than other forms of Tay-Sachs, and usually is initially seen in children between 2 and 10 years old. People with Tay-Sachs disease develop cognitive and motor skill deterioration, dysarthria, dysphagia, ataxia, and spasticity. Death usually occurs between the age of 5 to 15 years. Adult late-onset Tay-Sachs disease. A rare form of this disease, known as adult-onset or late-onset Tay-Sachs disease, usually has its first symptoms during the 30s or 40s. In contrast to the other forms, late-onset Tay-Sachs disease is usually not fatal as the effects can stop progressing. It is frequently misdiagnosed. It is characterized by unsteadiness of gait and progressive neurological deterioration. Symptoms of late-onset Tay-Sachs which typically begin to be seen in adolescence or early adulthood include speech and swallowing difficulties, unsteadiness of gait, spasticity, cognitive decline, and psychiatric illness, particularly a schizophrenia-like psychosis. People with late-onset Tay-Sachs may become full-time wheelchair users in adulthood. Until the 1970s and 1980s, when the disease's molecular genetics became known, the juvenile and adult forms of the disease were not always recognized as variants of Tay-Sachs disease. Post-infantile Tay-Sachs was often misdiagnosed as another neurological disorder, such as Friedreich's ataxia. Genetics. Tay-Sachs disease is an autosomal recessive genetic disorder, meaning that when both parents are carriers, there is a 25% risk of giving birth to an affected child with each pregnancy. the affected child would have received a mutated copy of the gene from each parent. Sachs results from mutations in the HEXA gene on human chromosome 15, which encodes the alpha subunit of beta-N-acetyl hexosaminidase, a lysosomal enzyme. By 2000, more than 100 different mutations had been identified in the human HEXA gene. These mutations have included single base insertions and deletions, splice phase mutations, missense mutations, and other more complex patterns. Each of these mutations alters the gene's protein product, sometimes severely inhibiting its function. In recent years, population studies and pedigree analysis have shown how such mutations arise and spread within small founder populations. Initial research focused on several such founder populations, Ashkenazi Jews. A 4-base pair insertion in exon 11 results in an altered reading frame for the HEXA gene. This mutation is the most prevalent mutation in the Ashkenazi Jewish population, and leads to the infantile form of Sachs disease. Cajun. The same 1278 INSTATC mutation found among Ashkenazi Jews occurs in the Cajun population of southern Louisiana. Researchers have traced the ancestry of carriers from Louisiana families back to a single founder couple not known to be Jewish or who lived in France in the 18th century. French Canadians. 2 mutations, unrelated to the Ashkenazi Cajun mutation, are absent in France but common among French Canadians living in eastern Quebec and Acadians from the province of New Brunswick. Pedigree analysis suggests the mutations were uncommon before the late 17th century. In the 1960s and early 1970s, when the biochemical basis of Sachs disease was first becoming known, no mutations had been sequenced directly for genetic diseases. Researchers of that era did not yet know how common polymorphisms would prove to be. The Jewish fur trader hypothesis, with its implication that a single mutation must have spread from one population into another, reflected the knowledge at the time. Subsequent research, however, has proven that a large variety of different HEXA mutations can cause the disease. Because Sachs was one of the first genetic disorders for which widespread genetic screening was possible, it is one of the first genetic disorders in which the prevalence of compound heterozygosity has been demonstrated. Compound heterozygosity ultimately explains the disease's variability, including the late onset forms. The disease can potentially result from the inheritance of 2 unrelated mutations in the HEXA gene, one from each parent. Classic infantile Sachs disease results when a child has inherited mutations from both parents that completely stop the biodegradation of gangliosides. Late onset forms occur due to the diverse mutation basia. People with Sachs disease may technically be heterozygotes, with 2 differing HEXA mutations that both inactivate, alter, or inhibit enzyme activity. When a patient has at least one HEXA copy that still enables some level of hexosaminidase activity, a later onset disease form occurs. When disease occurs because of 2 unrelated mutations, the patient is said to be a compound heterozygote. Heterozygous carriers show abnormal enzyme activity, but manifest no disease symptoms. This phenomenon is called dominance. The biochemical reason for wild-type alleles dominance over non-functional mutant alleles in inborn errors of metabolism comes from how enzymes function. Enzymes are protein catalysts for chemical reactions. As catalysts, they speed up reactions without being used up in the process, so only small enzyme quantities are required to carry out a reaction. Someone homozygous for a non-functional mutation in the enzyme encoding gene has little or no enzyme activity, so will manifest the abnormal phenotype. A heterozygote has at least half of the normal enzyme activity level, due to expression by the wild-type allele. This level is normally enough to enable normal function and thus prevent phenotypic expression. Pathophysiology. Sachs disease is caused by insufficient activity of the enzyme hexosaminidase. Hexosaminidase is a vital hydrolytic enzyme, found in the lysosomes, that breaks down glycolipids. When hexosaminidase is no longer functioning properly, the lipids accumulate in the brain and interfere with normal biological processes. Hexosaminidase specifically breaks down fatty acid derivatives called gangliosides. These are made and biodegraded rapidly in early life as the brain develops. Patients with carriers of Sachs can be identified by a simple blood test that measures hexosaminidase activity. The hydrolysis of GM2 ganglioside requires 3 proteins. 2 of them are subunits of hexosaminidase. The third is a small glycolipid transport protein, the GM2 activator protein, which acts as a substrate-specific cofactor for the enzyme. Deficiency in any one of these proteins leads to ganglioside storage, primarily in the lysosomes of neurons. Sachs disease occurs because a mutation inherited from both parents deactivates or inhibits this process. Most Sachs mutations probably do not directly affect protein functional elements. Instead, they cause incorrect folding or disable intracellular transport. Diagnosis: in patients with a clinical suspicion for Sachs disease, with any age of onset, the initial testing involves an enzyme assay to measure the activity of hexosaminidase in serum, fibroblasts or leukocytes. Total hexosaminidase enzyme activity is decreased in individuals with Tay-Sachs as is the percentage of hexosaminidase A. After confirmation of decreased enzyme activity in an individual, confirmation by molecular analysis can be pursued. All patients with infantile onset Sachs disease have a cherry red macula in the retina, easily observable by a physician using an ophthalmoscope. This red spot is a retinal area that appears red because of gangliosides in the surrounding retinal ganglion cells. The choroidal circulation is showing through red in this foveal region where all retinal ganglion cells are pushed aside to increase visual acuity. Thus, this cherry red spot is the only normal part of the retina. It shows up in contrast to the rest of the retina. Microscopic analysis of the retinal neurons shows they are distended from excess ganglioside storage. Unlike other lysosomal storage diseases, hepatosplenomegaly is not seen in Sachs. Prevention: 3 main approaches have been used to prevent or reduce the incidence of Sachs. Prenatal diagnosis: if both parents are identified as carriers, prenatal genetic testing can determine whether the fetus has inherited a defective gene copy from both parents. Couples are informed and may choose to have an abortion. Chorionic villus sampling, the most common form of prenatal diagnosis, can be performed between 10 and 14 weeks of gestation. Amniocentesis is usually performed at 15 to 18 weeks. These procedures have risks of miscarriage of 1% or less. Preimplantation genetic diagnosis: by retrieving the mother's eggs for in vitro fertilization, it is possible to test the embryo for the disorder prior to implantation. Healthy embryos are then selected and transferred into the mother's womb, while unhealthy embryos are discarded. In addition to Sachs disease, preimplantation genetic diagnosis has been used to prevent cystic fibrosis and sickle cell anemia among other genetic disorders. Mate selection: in Orthodox Jewish circles, the organization Der Yeshorim carried out an anonymous screening program so that couples with Sachs or another genetic disorder can avoid conception. Management: there is currently no cure or treatment for Sachs disease. Even with the best care, children with infantile Sachs disease die by the age of 4. Although experimental work is underway, no current medical treatment of the root cause yet exists. Patients receive supportive care to ease the symptoms or extend life. Infants are given feeding tubes when they can no longer swallow. Improvements in life-extending care have somewhat lengthened the survival of children with Sachs disease, but no current therapy is able to reverse or delay the disease's progress. In late-onset Tay-Sachs, medication can sometimes control psychiatric symptoms and seizures, although some medications are associated with significant adverse effects. In 2011, researchers discovered that pyrimethamine can increase hexosaminidase activity, thus slowing down the progression of late-onset Sachs disease. Epidemiology: Ashkenazi Jews have a high incidence of Sachs and other lipid storage diseases. In the United States, about 1 in 27 to 1 in 30 Ashkenazi Jews are a recessive carrier. The disease incidence is about 1 in every 3,500 newborn among Ashkenazi Jews. French Canadians and the Cajun community of Louisiana have an occurrence similar to the Ashkenazi Jews. Irish Americans have a 1 in 50 chance of being a carrier. In the general population, the incidence of carriers as heterozygotes is about 1 in 300. The incidence is approximately 1 in 320,000 newborns in the general population in United States. 3 general classes of theories have been proposed to explain the high frequency of Sachs carriers in the Ashkenazi Jewish population: heterozygote advantage. When applied to a particular allele, this theory posits that mutation carriers have a selective advantage, perhaps in a particular environment. Reproductive compensation. Parents who lose a child because of disease tend to compensate by having additional children to replace them. This phenomenon may maintain and possibly even increase the incidence of autosomal recessive disease. Founder effect. This hypothesis states that the high incidence of the 1278 INSTATC chromosomes is the result of an elevated allele frequency that existed by chance in an early founder population. Sachs disease was one of the first genetic disorders for which epidemiology was studied using molecular data. Studies of Sachs mutations using new molecular techniques such as linkage disequilibrium and coalescence analysis have brought an emerging consensus among researchers supporting the founder effect theory. History. Waren Tay and Bernard Sachs, 2 physicians, described the disease's progression and provided differential diagnostic criteria to distinguish it from other neurological disorders with similar symptoms. Both Tay and Sachs reported their first cases among Jewish families. Tay reported his observations in 1881 in the first volume of the Proceedings of the British Ophthalmological Society, of which he was a founding member. By 1884, he had seen 3 cases in a single family. Years later, Bernard Sachs, an American neurologist, reported similar findings when he reported a case of arrested cerebral development to other New York Neurological Society members. Sachs, who recognized that the disease had a familial basis, proposed that the disease should be called amaurotic familial idiocy. However, its genetic basis was still poorly understood. Although Gregor Mendel had published his article on the genetics of peas in 1865, Mendel's paper was largely forgotten for more than a generation, not rediscovered by other scientists until 1899. Thus, the Mendelian model for explaining Sachs was unavailable to scientists and doctors of the time. The first edition of the Jewish Encyclopedia, published in 12 volumes between 1901 and 1906, described what was then known about the disease. It is a curious fact that amaurotic family idiocy, a rare and fatal disease of children, occurs mostly among Jews. The largest number of cases has been observed in the United States, over 30 in number. It was at first thought that this was an exclusively Jewish disease, because most of the cases at first reported were between Russian and Polish Jews. But recently there have been reported cases occurring in non-Jewish children. The chief characteristics of the disease are progressive mental and physical enfeeblement, weakness and paralysis of all the extremities, and marasmus, associated with symmetrical changes in the macula lutea. On investigation of the reported cases, they found that neither consanguinity nor syphilitic, alcoholic, or nervous antecedents in the family history are factors in the etiology of the disease. No preventive measures have as yet been discovered, and no treatment has been of benefit, all the cases having terminated fatally. Jewish immigration to the United States peaked in the period 1880–1924, with the immigrants arriving from Russia and countries in Eastern Europe. This was also a period of nativism in the United States. Opponents of immigration often questioned whether immigrants from Southern and Eastern Europe could be assimilated into American society. Reports of Sachs disease contributed to a perception among nativists that Jews were an inferior race. In 1969, Shintaro Okada and John S. O'Brien showed that Sachs disease was caused by an enzyme defect. He also proved that Sachs patients could be diagnosed by an assay of hexosaminidase A activity. The further development of enzyme assays demonstrated that levels of hexosaminidases A and B could be measured in patients and carriers, allowing the reliable detection of heterozygotes. During the early 1970s, researchers developed protocols for newborn testing, carrier screening, and prenatal diagnosis. By the end of 1979, researchers had identified 3 variant forms of GM2 gangliosidosis, including Sandhoff disease and the AB variant of GM2 gangliosidosis, accounting for false negatives in carrier testing. Society and culture. Since carrier testing for Sachs began in 1971, millions of Ashkenazi Jews have been screened as carriers. Jewish communities embraced the cause of genetic screening from the 1970s on. The success with Sachs disease has led Israel to become the first country that offers free genetic screening and counseling for all couples and open discussions about the proper scope of genetic testing for other disorders in Israel. Because Sachs disease was one of the first autosomal recessive genetic disorders for which there was an enzyme assay test, it was intensely studied as a model for all such diseases, and researchers sought evidence of a selective process. A continuing controversy is whether heterozygotes have or had a selective advantage. The presence of 4 different lysosomal storage disorders in the Ashkenazi Jewish population suggests a past selective advantage for heterozygous carriers of these conditions. This controversy among researchers has reflected 3 debates among geneticists at large: dominance versus overdominance. In applied genetics, this controversy has reflected the century-long debate over whether dominance or overdominance provides the best explanation for heterosis. The classical balance controversy. The classical hypothesis of genetic variability, often associated with Hermann Muller, maintains that most genes are of a normal wild type, and that most individuals are homozygous for that wild type, while most selection is purifying selection that operates to eliminate deleterious alleles. The balancing hypothesis, often associated with Theodosius Dobzhansky, states that heterozygosity will be common at loci, and that it frequently reflects either directional selection or balancing selection. Selectionists versus neutralists. In theoretical population genetics, selectionists emphasize the primacy of natural selection as a determinant of evolution and of variation within a population, while neutralists favor a form of Mota Kimura's neutral theory of molecular evolution, which emphasizes the role of genetic drift. Research directions. Enzyme replacement therapy. Enzyme replacement therapy techniques have been investigated for lysosomal storage disorders, and could potentially be used to treat Sachs as well. The goal would be to replace the non-functional enzyme, a process similar to insulin injections for diabetes. However, in previous studies, the HEXA enzyme itself has been thought to be too large to pass through the specialized cell layer in the blood vessels that forms the blood-brain barrier in humans. Researchers have also tried directly instilling the deficient enzyme hexosaminidase A into the cerebrospinal fluid which bathes the brain. However, intracerebral neurons seem unable to take up this physically large molecule efficiently even when it is directly by them. Therefore, this approach to treatment of Sachs disease has also been ineffective so far. Jacob sheep model. Sachs disease exists in Jacob sheep. The biochemical mechanism for this disease in the Jacob sheep is virtually identical to that in humans, wherein diminished activity of hexosaminidase A results in increased concentrations of GM2 ganglioside in the affected animal. Sequencing of the HEXA gene cDNA of affected Jacob's sheep reveal an identical number of nucleotides and exons as in the human HEXA gene, and 86% nucleotide sequence identity. A missense mutation was found in the HEXA cDNA of the affected sheep. This mutation is a single nucleotide change at the end of exon 11, resulting in that exon's deletion via splicing. The Sachs model provided by the Jacob sheep is the first to offer promise as a means for gene therapy clinical trials, which may prove useful for disease treatment in humans. Dr. Edwin Kolodny and Fred and Joan Horak, Substrate Reduction Therapy, Other experimental methods being researched involve substrate reduction therapy, which attempts to use alternative enzymes to increase the brain's catabolism of GM2 gangliosides to a point where residual degradative activity is sufficient to prevent substrate accumulation. One experiment has demonstrated that using the enzyme silidase allows the genetic defect to be effectively bypassed, and as a consequence, GM2 gangliosides are metabolized so that their levels become almost inconsequential. If a safe pharmacological treatment can be developed one that increases expression of lysosomal silidase in neurons without other toxicity then this new form of therapy could essentially cure the disease. Another metabolic therapy under investigation for Sachs disease uses miglostat. This drug is a reversible inhibitor of the enzyme glucosylceramide synthase, which catalyzes the IRST step in synthesizing glucose-based glycosphingolipids like GM2 ganglioside. Increasing I-squared hexosaminidase A activity, as Sachs disease is a deficiency of I-squared hexosaminidase A, by getting a substance that increases its activity, people affected will not be deteriorating as fast or not at all. While for infantile Sachs disease, there is no I-squared hexosaminidase A, so then the treatment would be ineffective. However, for people affected by late-onset Sachs disease, they still have I-squared hexosaminidase A. The drug pyrimethamine has been shown to increase activity of I-squared hexosaminidase A. However, the increased levels of I-squared hexosaminidase A still fall far short of the desired 10% of normal HEXA, above which the phenotypic symptoms begin to disappear. References. External links, Gene Reviews NIHUW entry on hexosaminidase A deficiency, Sachs disease, NINDS Sachs disease information page, Sachs disease at NLM Genetics home reference, Sachs on NCBI.